you have this nitrate and phosphate coming up, and you have blooms in places where there's iron availability around islands and from the peninsula and so on. They have big blooms, and they take up nitrate, phosphate, and they also take up silica. But in the rest of the Southern Ocean, the silica is taken up despite the nitrate and phosphate not being taken up. So there's an excess of nitrate and phosphate, the silica is gone. Where does that silica go if there's no nitrate and phosphate? And this is what we showed. This alga, the one that grew, that they found 2 weeks later in Soiree, 4 weeks after the experiment, this Fragilariopsis, this is a diatom which has a very different strategy to those diatoms that make the blooms. This is a diatom that tries to persist in the surface layer. That's its evolutionary strategy. It's been selected for that. It has very thick frustules, very powerful, very strong. And we carried out here from the Alfred Wegener Institute, together with other institutions in the Helmholtz community, we carried out micro manipulation and experiments with shells. We tested the strength. And we found that this Fragilariopsis shells, the strength of the shell is so strong that if you were to take it up to table size for us to relate to, you could have an elephant stand on one of these shells, just to give you an impression of how strong they are. So this is of great interest to biocomposite people because the strength comes from elasticity. I can talk for hours about this aspect, the material properties. Anyway, so this shell is extremely strong. It's made to be very strong. And what happens is that this particular species is so strong that it's also swallowed by copepods whole. And it goes through the gut and can also emerge at the end and come out with the feces, and it's alive. So it survives gut passage. Not all, the majority do get crushed. By the way, a paper just came out now. This interests me a lot. I'm very happy to see it. These are Danish colleagues with whom I've been interacting. And they showed that the teeth of these copepods have teeth like that, right? With which they crush these diatom shells. And they showed that there were copepods that they fed with diatoms, and then with other organisms, like the one behind your shoulder, that Dinophysis. And the ones that fed on Dinophysis, their teeth were intact. And the ones that fed on these heavily silicified diatoms, they had broken their teeth. You clearly see that there were pieces broken off. So for them, it's the level at which they can crack shells is reached over there, right? And you all know there's a saying that you don't look a gift horse in the mouth. You also have that in German. Why? Because the teeth get ground down by the silica in the grass. So grasses protect themselves also with silica, just like diatoms. And they just have pieces of silica. They don't have a shell out of silica. They just have pieces of silica in the grass. And that's where you cut yourself on. You can cut yourself on grass. I'm sure you have. And particularly this, what we call pampas grass, is very tough. And that's because of silica. That silica protects the organic matter, and it wears down the teeth of herbivores, including horses. Right? That's why horses become old, because their teeth are down. And the cows and horses and so on, they only live for about 20, 25 years because their teeth are ground down by that time. So how do elephants do it? How do elephants do it? Elephants live much longer. They live as long as humans. But you know that elephants also eventually die of starvation because their teeth are also then flattened. The old elephants, they don't die of old age in that sense, just old age weakness like I'm headed to, but still some time off. But they die before that. And the reason why their teeth last that long is because they replace them. So elephants, in the course of their life, they get new teeth to replace them. So I just wanted to have this thing. Nobody had done this work before, looked at the teeth of copepods to see what effect the diatoms have, and they showed that it does. So again, we are on the same page with grasses and diatoms and copepods and elephants. Anyway, so these things all come together. Like, for instance, this map that you're just showing here, the Southern Ocean, the northern half of this map, of the green belt around the Antarctica in the south, only half of that would take up silica, would take up carbon in a meaningful way, because the silicate concentrations become limiting after the middle of the year, meaning after November, December, the silicate is gone, and there's a lot of nitrate and phosphate left, but then you don't get diatom blooms. This is what we showed with the experiment low effects, that there was no transport. So unless you have silica, you're not going to have deep carbon sequestration. That is only done by diatoms. And I think we know that now for certain. There's very little other phytoplankton that are taking carbon down to depth that don't have silica. And there's a lot of confusion in that sector, but these things are unknown now. There's data to support all these ideas. In the Southern Ocean, this Fragilariopsis I was telling you about is growing all the time. It's always there, and it's dominant, and it just sucks up the silica, makes its shells, and it also picks up iron when it goes through the guts of copepods, for instance, and krill, survives krill even feeding. Not all of them, just a few survive, but that's enough, because they come out and then they have picked up all that iron in the gut, and then they can divide like crazy for days, perhaps even weeks in the Southern Ocean. Yeah, but that's not enough for carbon sequestration. So the high silicate concentrations in the Antarctic are in the deep green region south, along the between the continent and there, right? And the other belts, there you don't get anything. It's no silica, because the silica is needed for building. You're making walls out of it. You can't have a thin wall, then it's not a wall. It has to have a mechanical function, and for it to perform a mechanical function, you have to have a certain amount, minimum amount. And that minimum amount is somewhere like between 5 and 10 millimole of silicate to make a decent diatom wall. And then once that's gone into the wall, it's gone. Because that silicate takes a long time to dissolve out of the frustule, weeks.
Now, here you see now the buildup of these blooms. So the black squares or rhombuses are inside the patch, and the open white circles are outside the patch. So you see the Pseudonychia lineola, this is the species. It took off in the bloom, in the EisenEx bloom, after adding the iron. Whereas outside, nothing happened. And this is another species, Chitoceros corvizatus, that also built up biomass. No? And you can see how they started growing. And this went into exponential. So it had made a big bloom. Now, Pseudonychia is a genus which is known to sink out. It's not eaten much because of these chains of needle-shaped chains. They are not appreciated by the average copepods, so it's not eaten much. But of course, all sorts of other phytoplankton also grew. But here you see these two that responded with an exponential growth. But this Pseudonychia started at higher concentrations, right? Here's the biomass milligram carbon per square meter. It went up to 1 gram, right? We had 1 gram of biomass per square meter, and Chitoceros was just 1/10 of that, because it started with lower concentrations. Now, this Chitoceros, this particular species here, but also other species of Chitoceros, they make resting cells that are like spores that are very different to the vegetative cells. And these are seeding. These seed phytoplankton blooms. So this is in the open ocean, and these are actually classic coastal species, at least in today's ocean. Because the resting spores settle out at the sediment surface, and when that sediment is remobilized, is resuspended in the winter, or by storms and so on, it then comes up, and then they can germinate and grow. And I think that if we want to have a big carbon drawdown, we should target these species and get spores of these species to populate the water and build up biomass, because they are also very well-known sinkers. No? They make big blooms, and when the blooms are over, the seasonal cycle, then they make aggregates and they sink out. And they take these spores with them. Now, this is the copepod story. Look, here you see the red is outside and the black is inside. And these are copepodites and adults in the patch. And so we started off with same values, but then we did have higher. The copepodites are the juvenile stages, So it could be that these juvenile stages grew faster. So we had more of them because it went from nauplii to copepodites faster. But this is very significant. We had a significant boost in the zooplankton. And then if you look at another tracer that we followed, and this is the incidence of broken diatom frustules. Right? So you count the diatoms, and I showed you some pictures of broken diatom frustules, and then you can see that this has been grazed. Right? You count how many phytoplankton cells were intact and how many were broken, because the breakage is only possible with copepod mandibles. There's no other way to break frustules. So if you have broken frustules, it means that this is broken by a copepod and ingested and then defecated and then spread out in the water column. So here you see inside the patch and outside the patch the incidence of broken diatom frustules and significantly more breakage inside the patch. So the grazing pressure increased. The zooplankton were feeding more inside the patch, much more. So this was the EisenEx experiment. Of course, you got a lot of more data on all the other organisms, but I won't go into that now. And published this in Nature, deep carbon export from a Southern Ocean iron fertilized diatom bloom. And this is the bloom we had. Now, this is Chitoceras, the different species of Chitoceras. And these are aggregates. And this is inside the patch, and this is outside the patch. And these are tintinids, ciliates. No, I told you about tintinids that have a shell made out of. Do you remember? Does anybody remember what the shell of tintinids is? Yeah, excellent. Yeah, very good. The skeleton sinks out. No? Same skeleton as your fingernails, right? And your hair. They have the same compound. And it sank out. And here inside the patch, you had all these diatoms. So the keratin carrying ciliates, they were inside and outside the patch in similar quantities. But inside the patch, we also had these diatoms. So these are all the diatoms. And here you can see the size relationship. And then at a certain time, after 3.5 weeks, the bloom peaked and it sank out. And these are the aggregates that were formed by the diatoms. And here, this blue stain, it's cyan blue, it stains acidic polysaccharides. And these polysaccharides, they are binding, they're sticky. So they bind cells to each other. And here you can see the stuff oozing out of this cell. So this cell is dead, and its contents have been broken down, autolyzed. The enzymes inside the dead cell, they start working, breaking down. Now, before that, they were building up, but when the cell is disorganized, it breaks down. And then this material, this slimy material, comes out. I don't know whether you all have ever had the experience of putting salad into a freezer or something, and the cells break down and it all becomes slimy, right? Or if you cool something too much, then it becomes slimy. That's autolyzed plasma, right? That's plasma of that whatever, the leaf or whatever, that then where the cell walls all break and the enzymes go wild because they're released. And that's called autolysis. Self-lysis is breaking down, and autolysis is when you break down yourself. And that's your own enzymes break you down, so to speak, right? So we have this in diatom blooms. When they reach a certain peak, when they reach their peak, then they go into this autolysis, and then you have aggregate formation. The aggregates I showed you in this previous slide, no? Then you get this. No, sorry. Yeah, then you get these aggregates. Now, this is the patch. This is from that same paper I showed you just now. So this black dot is the fertilizer. Right? So this is the map that we made of the entire eddy. Right? So it's an eddy going around this direction, clockwise, and in the Southern Ocean, you have the Coriolis effect going to the left, so this water is being pushed out of the center of the eddy, right? So that means this cold water, so to speak, comes up, right? That's why it's a cold core eddy, because it's pushing the surface water out. But of course, we also measured that. We measured how much water was coming up. It was negligible. It's just that it maintains that this cold core here. And you can see the cold core here. This is the sea surface height anomaly, SSHA. sea surface height anomaly. And this is the first round that the patch made. We unfortunately didn't hit the middle of the patch. We fertilized, you can see over here, we missed the core, but then this patch went around this direction and it came over here. Then we took the next measurements. And this is the chlorophyll. So this is like 1 week after, this is the first round. This is 1 week after the patch. This patch went around, and then this is how it spread. And the chlorophyll, the amount of chlorophyll that was then produced. By the way, this paper is on the web. It's not behind a paywall, so you can, if you're interested, you can access it. In Nature, 2012. Now, this is the FV over FM, and this is the second round. Right? This patch went around 3 times inside the eddy. This is the second round, and what we did over here, we went to this place here, and then we just did sections, up and down, up and down. We just went north-south sections and let the patch go under the ship, go around. Right? We didn't follow it. We just did this, and the patch moved under it. And that's what you can see. Now, this is the extent of the patch, and this FV over FM is the photosynthetic efficiency I told you about, measured with the RRF fluorometer on deck. And this is the CO2 concentration in microatmospheres. And here, you see the scale. So the red is high values, and the blue is low values. And that inside the patch, we had 4 microatmospheres. This is equivalent to ppm. This is almost the same as ppm in the atmosphere. So we went from 380 ppm, this is the high values, down to 344. They were taken up by the second week by this patch. Yeah? And this is the chlorophyll. So this is where the chlorophyll was concentrated, and in the patch. And these are satellite images. So this is before fertilization on the 2nd to 12th February, and here, this is the eddy. So you see that inside the eddy, there was less chlorophyll. But outside the eddy, there was a band of chlorophyll. Right? But then, this is on the 12th of February, and this is the 26th of February, 1 month later, or a bit less, to the 11th March, about a month later. Here you see our patch. Right? This is what we had fertilized, how it has grown, but the diatoms around the patch, they had gone. They had sunk out, because that's what normally happens in the Southern Ocean. Diatoms reach a peak and then they sink out. And then this is again 1 month later, and then we still see a bit, but the reason why these pictures are not so good is because of cloud cover. Now, you don't get rarely get a good image of the sea surface, because you have to go through, looking through the clouds. That's why it's over days. No? This is 2nd to 12th February. Whatever values we got from holes in the clouds were then assimilated into this one picture. Yeah? Second round. And so this is the patch. Now we're going to look at the water column, taken with the CTD, with these water bottles that you then lower into the water. So this is the chlorophyll concentrations going from below 0.5, this is the low values here, up to above 3. Right? So these are the highest values we got here after the days are over here. No? From day -1, this is before fertilization. This is the station we took before adding the iron. And then we added the iron, and then you see already here, this is out patch. In patch, in this period, we went and did the other studies. And that outside the patch, nothing much happened, but inside the patch, you had this buildup of the bloom. So you can see how long it took for biomass to build up. And after 3 weeks here, this is when the bloom more or less peaked. And then it reached high values, and then it started going down again. And this is when we left. This is after day 35 something. That was the end of the experiment. So 7 weeks we were over there. And the experiment lasted 6.5 weeks, because it includes the pre-patch. So this is how the chlorophyll built up, and you see it goes down to 100 meters. So this idea that I had that the mixed layer prevents phytoplankton from growing, rubbish. Because we could show that 100-meter mixed layer, we built up this huge bloom, right? what I mentioned yesterday, or when we were talking about phytoplankton, I mentioned that this critical depth hypothesis, it's not crucial for understanding phytoplankton ecology. So I don't teach it anymore. It's a bit of a red herring. Yeah? And it's taking time, but it's being accepted. That's not crucial. Yeah. Now the other interesting thing, these are silicate concentrations here, silicic acid. And here you see now the uptake, how silicic acid is taken up by the bloom. Now this purple are low values, below 10, and the orange is above 40. So at depth, you had high silicate concentration. You're in the Southern Ocean. It's very high normally. And here, this is inside the patch. But you see the silicate being taken up by the diatoms inside the patch. Now, the shocking thing was that this also happened outside the patch. There was no buildup of chlorophyll outside, but the silicic acid went. It was still being taken up. Right? Now, here you see this is EisenEx, the experiment that we had to stop on day 21, one of the saddest days in my life. And this is EIFEX. So we extended the phytoplankton, then you see much more chlorophyll buildup, and then also the decline at the end, when this bloom was going down. And here you see the depth of the mixed layer was down here, and they still built up so much biomass. Now this is a little bit trickier. What comes now? These are integrated values of all the important parameters, right? POC. Remember what is POC? Exactly. Particulate organic carbon. This is particulate organic nitrogen. This is particulate organic phosphorus, phosphate. This is biogenic silica, right? This is particulate. This is silica in particles in the diatom frustules. It's called biogenic silica. The other one is dissolved silica. Biogenic silica, this is chlorophyll, and this is ammonium on this side. And okay, I'm sorry, I'm slightly confused. This is dissolved inorganic carbon, this is particulate organic carbon. This is nitrate and nitrite, this is particulate nitrogen, organic nitrogen in the particles. This is dissolved phosphate, this is organic phosphate, particulate organic phosphate, yeah. So this is in the particles. This is silicic acid, this is the dissolved silica, this is the biogenic silica. This is ammonium, this is chlorophyll, this is dissolved organic nitrogen, and this is dissolved organic carbon. Right? So these are the individual measurements over the course of this experiment. So you see how the dissolved inorganic carbon, right? This is CO2 and bicarbonate, and well, the dissolved inorganic carbon, how it went down inside the patch and stayed more or less constant outside the patch. Slight reduction. Particulate organic carbon went up, because this is the carbon that was taken out of the water appeared now as particles. Right? You remove the dissolved, and that dissolved goes into particles. And then at the end, this particulate organic carbon went down. So this going down is due to sinking out. Get it? The particles are sinking out. So you have it goes down. Now we saw the same thing in the nitrate and nitrite. No, they went down. And the organic nitrogen went up, particulate organic nitrogen. And outside the patch, you had a slight decline. Yeah, so you're understanding now this transition from dissolved to particulate. This is uptake by the phytoplankton. And the data, of course, messy, because each time we had to leave the patch and then go back again and find the center. And that patch is spreading and is moving around, so it's a bit messy. I mean, you can't expect really clear-cut data distributions like you would in a culture experiment, right? But even in culture experiments, you don't get things really clearly, because you lose stuff between the dissolved and the particulate. Now, this is a big problem we had. Phosphate went down very nicely, but then we had these high values over here. Then we couldn't explain them. Why the hell did the phosphate go up again? And we checked the data. That's what we found. There was no way, there was no contamination or this, that, or the other. But so that remains a kind of question mark. Why did the phosphate values go up again towards the end of the experiment? The only explanation I could have is that these diatoms were bursting, right? I showed you those autolysis, and that phosphate was released again. And not the nitrogen, right? The nitrate, you didn't see that behavior in the nitrate. But of course, you wouldn't expect it here. But you wouldn't also expect it in the ammonium. Or you could. You could see that this increase in ammonium here could be due to the release of ammonium from the dead cells. No? Because then the proteins are broken down by proteases that are in the plasma, and then that's released then as ammonium, right? And the really surprising thing we had was this dissolved organic nitrogen. Now, measuring dissolved organic nitrogen is very tricky. But up here, you can see that the concentrations were well above 42. The millimole per cubic meter of dissolved organic carbon. Now, the nitrogen, you see, it went down inside and outside the patch. But there was no comparable increase in the particulate organic nitrogen here, right? So this went down by about from around 4 millimole down to about 2, whereas here you had an increase from 1 to 2.5, right? So this decrease here explains this, right? But yeah, so I doubt these measurements. You know, that they were measured in Southampton in another institute. No? This is a European experiment. So perhaps they're not really correct. But of course, you can't then just put it in a drawer and forget it, because you don't have an explanation why they're incorrect. If you know that this is contamination of something, then you can say, all right, I'm not going to show these data. But if you have no explanation for why, you have to show the data. You have to be totally honest. That's so important. I want to stress this point very much, that you should be always, when you have data, you should always assume that unless you can prove that the data, there's something wrong with the data, you have to face your data. Because my history of plankton ecology has always been facing the data, and I've always had success. And so I even put stuff which I'm not sure about, which I can't explain. Okay, I can't explain it now, but at least this is what we found. I mean, this is the data we got. You know, I have colleagues who say this doesn't fit, and then we put it in the drawer and we forget about it. But then you miss stuff. If you do that, you know, it could be that you found something sensational because it doesn't fit your mainstream thinking. Right? How are you going to find anything new if everything you find fits what you knew before the experiment? Yeah? So be honest with your data. That's really, really, really important. Generally, you know, you should be transparent and open and everything, you know, on the table. Discuss everything. Yeah, this is what we found. That's so important. We won't make progress otherwise. We will only stick to what we want to believe. And that becomes a religion. Right? Anyway, so you know, we show whatever we found, and okay, we can't explain it now, but that's what we found. But it doesn't contradict the main message. The main message is clearly here. The POC went up and then it went down. So where did it go? Where did it go when it went down? Now these are transmissometer profiles. This is down to the seafloor. We only went down to 3,000 meters. I mean, the CTD went down to the bottom, which is 3,700 meters here, which is the average depth of the ocean, 3,700 meters. So we went down to 3,700, but at depth, you always have some sediment resuspension. It's called the nepheloid layer. Nephelos is fog. The nepheloid layer means that there are particles in the water. And this is due to resuspension. So if you want to look at clean data, we have to then go, not go down all the way to the bottom. So not to confuse, we just look at the 3,000 meter water column. So this is on the beginning of the experiment, before the experiment was started. And this is the mixed layer, this is the plankton in the surface layer. And then on day 32, of course, we were taking the CTDs regularly. And one of the big regrets I had is that we didn't take more deep CTD casts. Because each CTD cast, down to the bottom, takes 3 hours. It takes 1.5 hours to go down 3.5 kilometers, right? You have these drums with wires, right? And you sit there and you know, but of course it's exciting because you're watching this going down and you see all this. See, these peaks you see here, these are bigger particles, right? And yeah, there's a lot to be found out over here in the deep ocean. So here you see how in the deep water column, these particles increased. First of all, you see at the top, then this is the plankton bloom, that you have more turbidity at the top, right, compared to the beginning. And here you see how much the particle load in the water column has increased. Right? By the way, this water that you have, let's say this depth over here, is so clear that you cannot make it in the lab. So these transmissometers cannot be calibrated in the laboratory because you just cannot get that clear water that you have in the ocean at depth. The water in the ocean, the deep ocean water, is so clear of particles that you cannot replicate it in the lab. Because there's always some contamination in the lab. And just to show you how clear that water is, I mean, it's all the particles have been either sunk out or they've been taken up, or they've been broken down. And if you get peaks like this, and this is mostly organisms responsible for that. Right? And here these are sinking particles, clearly, from the bloom. Right? Because this is day 32, 36. So this is 7 weeks after the starting the experiment. This one. So what we did, we then integrated, because, you know, so our CTD, whenever we put out the CTD, it went only down to 500 meters. Right? So the upper 500 meters was taken every time we put in the CTD. But these deep casts down to 3,700, they were scattered. And we only did that after we saw the bloom sinking. Then I ordered more of these deep CTDs, and I regret that we didn't take more before. So, yeah, so this bloom sinking, when we have all this data which clearly show this was going down. So, and then what happened is that we integrated this. Yeah, I have to go back again. So what I'm going to show you now is we are integrating the particles in these different layers, in the 100-meter layer, in the 100 to 200-meter layer, 200 to 300, 400, and so on, going down. Right? And these layers are now shown in this diagram. So this is the top layer. This is transmissometer, right? But it's taken into POC. Now POC are the red dots. These are water samples taken at that depth and measured in the CHN analyzer. And the black dots are the results of the transmissometer, right? Integrated. So you see, now this is inside the patch and this is outside the patch. And we only have one data point from below the patch in the deep water column, because this is 500 to 2,000. So we have a lot of 500-meter depths, but we only have these ones from the deep water column. They're taken later. And there's so many dots here because we had to put in the CTD for different samples, right? So, for instance, somebody was measuring the POC, each water bottle is 12 liters. And you have to apportion that water. So 12 liters, then that means that if you're filtering 2 liters of water for POC, 2 liters for chlorophyll, 2 liters for this, 2 liters for that, then that water bottle is finished, right? And also for nutrients and so on. And then you have all sorts of other people who are measuring all sorts of other parameters that I haven't shown over here, like for DMS, DMSP, for pigments, for kind of different types of pigments and so on. So, and for thorium, I'll show that also, that gives you a loss rate from the water column. So all these different, during the station, you have all these different casts of the CTD going down and taking these measurements. And that water is then taken for different measurements. That's why we have so many dots, right? It's all individual CTD casts from that station. So what are we seeing? So we are seeing now here the surface, you have this. Now this is very messy because this is 100 to 200 meters and the scale changes. Now here we go from 0.6 to 1.2. This is the surface, but here it's below 0.4. Right? It's from 0.5 to 0.2. And this is messy because it's at the base of the mixed layer. And that mixed layer is not constant, it's not flat, because you also have waves, internal waves in the ocean, where the surface layer is also, has a wave going through it at very long scales, kilometer kind of scales, because the ocean is not motionless, right? So then you get a mixture of the bottom of the mixed layer and then the top of it, right? So this you have to disregard because it's messy. We have to draw a point somewhere and say we're going to integrate at that way, standard. So this is not giving you much information. But below that, 200 to 300, you see clearly how in the course of the experiment there was this increase, right? And then you had the sudden rapid increase. And you see that here, 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 and here, and here. And the red dots are the samples that were taken. And the samples were taken from large water bottles because you have to filter a lot of water to get enough sample to make that reliable measurement. Yeah? And this is outside the patch. So what you see is that inside the patch is, forget this level. Here you had a decline. The phytoplankton went down, the concentration actually in the course of the experiment. And you had an increase at depth, meaning those particles went down. But remember, the scales are different. So this is much less than what went down there, actually. So you had this slight increase and outside the patch. And the grief that I got was from these values, these two values, right? These high values outside the patch at depth. So how can you prove that this is not due to this or the other? Yeah? So, yeah, I mean, there were blooms in the neighborhood, and there was some exchange, and so we did have these two values, higher values. Now this lot over here also outside the patch, and this is like lateral contamination. I mean, anyway, so that was a reason why we had problems proving that our patch was a result of the iron that we added. That these high values that we had here. Anyway, but I think that the evidence from this data is pretty overwhelming, that you had low sedimentation in the beginning, right? Low sinking out. There was a slight increase in the particles, but then from day 24 on, sort of here, then suddenly this thing increased, right? And then you have massive sinking out. Now what you can do is you can take all the data from these different days here and integrate them for the entire water column, right? And that's what we did here. This is the triangles are the 500-meter data, right? The 500-meter integrated, right? And these big black circles, they are deeper. No? So the in-patch 0 to 3,000 meters are these circles, right? We had only one value here, and then we had this here. And the square is an edge patch. This is an edge patch. This is an edge patch. Because later on we had to go through the data and find out which are inside the patch, which are outside the patch, without biasing, right? So this is inside the patch and this is outside the patch. And these are these three data that gave me grief, right? How did you get such high values outside the patch? Yeah, and I could just say that, well, there were particles hanging out there in the water column, all right? Now, here's the gram carbon per square meter, and moles, in moles. So if you, to get the gram carbon, you have to multiply by 12. And here you see that if this is the base, if one is the base, 1 mole integrated for the water column, and here we went up to almost 3, so that's 2 moles, that's almost 20 gram carbon. Actually, but then we said 10, all right? We don't want to exaggerate. So we said that about 10 gram carbon per square meter sank out of this bloom. This is the stuff that appeared here in the deep water column, right? If you take away the 500 meter depth, right? So this bloom that we had sank out about this amount of carbon. The nitrate went down from 20 something, okay, it was about 2 or 3 millimoles of nitrate that were taken up. Because there was still a hell of a lot of nitrate in the water column when the bloom stopped, right? So it was iron limited. So if you had added more iron afterwards also, I think we would have prolonged the bloom and then we could have got 1 or 2 gigatons of carbon from iron fertilization like that, right? Okay, so here you see how we can put these things together, and here you see the actual results that we got. No? The difference between these values here and these values here, this is this, let's say one mole, that's 12 gram carbon. That is what definitely sank out of the water column, sank out of our patch. So looking at this diagram, you can follow the buildup of the bloom, right? Here with this in the upper 500 meters, and then you can see how the stuff sank out. So this kind of integrated picture of the entire water column, you can only get in an eddy, because the eddy is rotating from the top to the bottom in the same way. The entire eddy is revolving like a cylinder. So we could track the eddy down. We could track the particles down to the bottom. And that was an issue at the time, that to what extent are the particles sinking out of the surface layer that appearing in the deep water column. So all the other experiments, they just went somewhere and did the experiment, put the iron in the water, but it did not have a coherent water column, right? So this eddy acted like a test tube, right, in which you run the experiment. So if you do future experiments, they should be run in eddies, no? Because that's the best way of doing the experiment. I'll show you later on the low-effects eddy, where we didn't have as much luck as we had in this one. This one stayed coherent. All right, so now I'll look at some of the organisms that were involved in this build-up of biomass. This is inside, this is outside, and this is chlorophyll A and phytoplankton carbon. Now you see that chlorophyll A and phytoplankton carbon went together very closely. The phytoplankton carbon is calculated from cell counts. Right? The sample was taken and counted, the cells of the different species were counted under the microscope. And then not only the cells were counted, but also the broken frustules, fecal pellets, and whatever was there, whatever particles were there were also counted. Here you have the phytoplankton counts, which I just mentioned. No? This is outside, and this is inside. So you see the peak, and then you see the decline. You see the things went down, but then we found what declined, we found down there. So if that had stayed up here, no, it would have just gone on up to there, like in this previous diagram here, right? This is for the entire water column. This is the only time that this kind of data had been published, or recorded even, because the deep water column is always separate from the surface water column. And because we did this experiment in a cylinder, a coherent cylinder, we could get this. We had the total content of everything, the fate of everything and everything, right? So this was easily by far the most successful experiment up to now. So this is this Fragilariopsis kerguelensis. You see these empty cells, right? This is the ones I'm talking about that are sinking silica, but no carbon. Although the amount of silica that was taken out was a hell of a lot. I mean, we didn't really find that in the samples, but this silica, empty silica, is transparent to the transmissometer, because the transmissometer, the red light goes through the cells, through the empty frustules, glass. It's the carbon that gets absorbed. Anyway, so here, this is Fragilariopsis chains stained with DAPI. DAPI is a stain that covers DNA. So you see the cell nuclei very clearly. Right? And here there are no nuclei, that means those frustules are empty. And still the damn thing stays afloat. How it does, doesn't know. This is a light micrograph, also Fragilariopsis. This is with the chloroplasts. The same as this one. Yeah, and these species, this one and this one, they were the major sinkers in the in this experiment, right? And this is Thalassiothrix. This has very, very long. So this is wrong. It should be much smaller actually, because these are 2 millimeters long. These are the biggest diatoms in the world. This particular species, and they have very thick shells. I showed that when we were talking about the diatoms. Anyway, so this is what you saw. Now here's the chlorophyll measured in the laboratory after filtering the samples, and the blue is the phytoplankton carbon calculated from counting cells. These cells, they were all counted, and then the carbon, they were given carbon values, and that was multiplied, and then you get that accurate results. And this is then now the total biomass, and the black stars are the measured POC in the filters, the carbon. And here you see the green is the diatoms, the pale green are other phytoplankton, black are the bacteria, this pale, this salmon is protozoa, the red are the copepods, and the yellow are the big copepods. But again, the copepods are definitely underestimated because of the net escape. No? But here you get a feeling for the relative amount of carbon in these different categories. And then you also see who's increasing into what, right? So outside we had a lot of copepods, but we had less diatoms, right? So the reduction outside here, this reduction, was due to the diatoms going down. Probably grazed. Yeah? Whereas inside they increased. But the others didn't change much. Now the protozooplankton, for instance, stayed more or less similar, and as I told you, the bacteria are always constant. They're the same. Yeah? And here pseudonychia was minor in this experiment. It was a major in the other one, but in this one it was minor. So again, which species start growing is also variable, and we don't know exactly which. We can't predict that yet, because we don't know enough about them. So these are again different species. This is Chaetoceros decata, and you can see how it went up and went down. Now these here you have full cells, empty frustules, and broken frustules. So you can see that in the beginning there were not much grazed, but then there was more. This broken frustules means that something cracked them, copepods. Right? And empty means that the contents of the cells went out due to autolysis, apoptosis. Remember apoptosis? Programmed cell death, PCD. So then the cells become empty. So here you see this went up and then it went down, this one went up and went down. So these were the ones that contributed primarily to the fallout of carbon, no? The 1 gram carbon per square meter that sank out to the bottom. Here you see other species. Yeah, so here you can see them going down. Yeah, okay, this goes on. And this is from 350 meter depth. These are water bottles that were taken at 350 meter depth, and where the amount of particles, the particles were looked at under the microscope. And you see that there were none down there in the beginning, hardly any. And then, in the peak of the bloom, on day 32, 34, they appeared at 350 meter depth. Right? The same over here, this species, but not that much. But here you see actually the stuff going down. Yeah? Then we have this other group, Corythron inermi, and here you see they didn't sink out. This group didn't sink out. Rhizosolinia huni also didn't sink out. Yeah? And then you look at depth, you see that there was nothing at depth from these species because they didn't sink out. Right? So this is how you clarify this biological carbon pump, about which there's so much confusion and what is it doing, not doing, and so on, right? And it's pretty straightforward. These species did not sink in that time, and the others sank. They had a phase where they sank out. And we could see under the microscope also that they died and then sank out. We could see the dead ones. Yeah, so this is like day 0 when we started the experiment. This is a kind of overview of the plankton. This is at the height of the bloom. See all the Chaetoceros? And this is then at the end, on day 37. And you see that all these Chaetoceros have gone. They sank out quantitatively without taking the others out, right? So this is, again, not part of normal bioceanography knowledge, that the sinking is species-specific. Species, populations of specific species sink out, and the others stay behind. It's like with the trees. If you look at if you've been following the trees, no? Like this one here, this alder, is still green. The leaves are all green. And then there are many trees down there which are empty. All the leaves have fallen off. So we also have this kind of seasonality or phenology, it's called. No? Phenology is the life cycle of organisms. No? And the phenology differs from species to species. That's why they're species. They're not all the same. Yeah? So you see that here clearly. Now, by the time the ship left the experimental site, 37 days after fertilization, about 1 mole had sunk below 500 meter depth. In all likelihood, this amount had settled out in the seafloor. I mean, they went all the way down to the bottom. And the C to Fe ratio of the sequestration was at a minimum 15,000 to 1, but closer to 30,000 because there were still so many diatom species at the surface. What happened to them, we don't know because we left. But I'm sure that many of them also sank out, except for the Fragilariopsis and the Thalassius. Well, some species didn't. They died in the surface layer. They don't all have to die in the surface layer and sink out. Right? And here's this again, the Alcian blue stained aggregates. No? These are the aggregates. And here you see this, these are the different kind of the phytoplankton. You see this, the plasmolysis. No? You see the cell contents seeping out as a slime and picking up other stuff. But it didn't pick up every particle, by no means. So then, based on the results of this experiment, we published this paper. It's called Thick-Shelled Grazer-Protected Diatoms Decouple Ocean Carbon and Silicon Cycles in the R-Unlimited Antarctic Circumpolar Current. And you remember, yesterday I showed you the distributions of nitrate, phosphate, and silicate. And you saw that they were not tightly corrected. I mean, the nitrate and the phosphate were quite closely related. They were correlated with each other according to the Redfield ratio. But the silica, you had places where there was little silica, and there were places, you know, like in the Antarctic Circle, in the intermediate water, there was little silica going into the ocean, but a lot of silica was going down in the deep water, and so on. And that's due to this effect. It's due to the biology of these different species. So the ones that are thick-shelled, why do they make thick shells? Because they're grazer protected. So they can survive in the surface layer because they're defended. Whereas the thin-shelled ones, they have to go. Right? So you can start making life cycle strategies of the diatoms that has not yet been made. It should be done, but it can be best made with these fertilization experiments, because there you can follow them. Otherwise, you don't get that connection. Okay? Yeah, and then we also looked at the zooplankton. Now, this is a euphosid. There were also some euphosids there, not many. It was mostly copepods. And here you see this Fragilariopsis kerguelensis has gone through the gut. I think I showed this already, did I? Yeah, I did. Yeah, and when I was showing the diatoms. Yeah, it came from this experiment. So these cells are alive.